Hello out there in food space. You are here because you make or want to make a food ingredient through fermentation, in particular a protein or an enzyme. You might have a particular protein or enzyme in mind. You might have a function in mind, and you're looking to discover a protein that can perform that role. You might be looking to produce a very high volume ingredient that makes up a large percentage of your final product. Or you might need something in smaller quantities for the flavor, for the nutritional value, or for some other active property. Food proteins are diverse, but the underlying technology is similar because these are peptide sequences, they're coded in DNA. Here at Ginkgo Bioworks, we can design that DNA, transform it into a microbial production strain, and then create an engineered organism for you to use when you produce your protein by fermentation at scale. The goal of today's event is for us to lay out what Ginkgo can do for your food protein projects and describe some of the technology that makes it work. So what is on the agenda? First, I'll tell you about Ginkgo, who we are, the services that we offer. Next, we'll review some key pieces of foundry technology. These are the capabilities that we think allow us to execute your cell programming projects on a level that nobody else can. Then we're going to deep dive on two of our favorite expression hosts, Pichia and Aspergillus. We've got some case studies on how we engineer those hosts and how they performed specifically in food protein projects. Then comes a very special feature. We've recently started to roll out a success-based pricing as an option for certain protein and enzyme projects. This is what it sounds like. You only pay if the project works, but of course there are some important details that we'll review. And then finally, we'll open it up for Q&A. And for that, I'd like to call attention to the Q&A button at the bottom of your Zoom screen. You can click on that button at any time to ask a question to our team of experts. Could be a technical question, could be a question about doing business with us, anything at all. And at the end of the session, hopefully you will know everything you ever wanted to know about Ginkgo Bioworks. Speaking of which, let's talk about Ginkgo. We are over 1,200 people. We're based in Boston. We have offices in California, France, Netherlands, Switzerland. If you come to visit our headquarters, you'll see a lot of robotics, a lot of automation. And that's our foundry platform. Between the technology investments and the physical scale, we've put almost $500 million into building out this foundry. And we didn't do it so that we could develop our own functional proteins and our own food enzymes. We did it for you so that we can partner with you to make those products. A good way to think about our business model is we're a CRO. You can send us your R&D projects on a contract basis, but we're more than a traditional CRO in the sense that you come to us and you can get things that you can't get anywhere else. So what can Ginkgo do for you? Here's our menu of service options for proteins and enzymes. We do protein discovery. If you don't have a protein in mind, we can find you one. We do protein engineering. In a typical case, this means machine learning guided changes to the sequence and the structure to improve things like stability, expression, or functional performance. We do expression optimization. That means changes outside of the protein sequence itself, like the promoter, the host background, usually with the goal of improving titer. And we do fermentation scale-up. This service usually ends with a tech transfer package to your contract manufacturer, allowing them to ferment your optimized strain under optimized conditions. The big idea here is that we're integrated, we're full stack, so you can partner with us for one of these menu options or for all of them. And we find that that foundry integration is very powerful because it means we can invest in the kind of technologies that produce efficiencies and economies of scale. What kind of technologies? What makes us stand out? In the food space, I can summarize it like this. We've got DNA sequence libraries for discovery that are much larger than the public databases. Our customers get access to that resource for enzyme and protein discovery. We've got a machine learning team for enzyme and protein design with deep experience, dozens of projects under our belt. Equally important, we have foundry automation and high throughput assays for feeding data to that platform. Machine learning is only as good as the data you can feed it, and we do big data around here. We're going to showcase NCAPS, our encapsulation and screening platform. This is a microfluidic system that works at ultra-high throughput. We can test up to 1 million strain designs at once with NCAPS. And then the real stars of the show today are going to be our commercial production strains, in particular Pichia and Aspergillus. If there's one message that we're hoping to get out there with this event, it's that the strain you use matters. And at Ginkgo, we've got some high-performing production strains with a track record at commercial scale. We're very proud of these. They're proprietary. You can't get them anywhere else. Pichia and Aspergillus. Let me break it down here for you. Both of these have the key features that you need if you want to make a food ingredient. They're food safe, they're methanol free, they grow on low-cost feedstocks. Pichia tends to win for making more complex proteins. It's easier to engineer at scale. There's a larger genetic toolbox. So if your protein is hard to fold or if it has a fussy cofactor, you're probably thinking about Pichia. Aspergillus tends to win on sheer titer. For the proteins that Aspergillus does well, you can get really insane productivity. So that picture, maybe it's a little oversimplified, but you get the idea. The right strain for the right project, the right tech for the right project. And at Ginkgo, we want to offer all the right strains and the right tech. So now, to tell us more about the tech that Ginkgo brings to food, we welcome Sneha Srikrishnan. Sneha is a scientist with over a decade of experience delivering proteins for nutrition, wellness, and other applications. At Ginkgo, Sneha is a director of business development for products in the protein and enzyme space. Welcome, Sneha.
Thank you, Jake. Very excited to be here and talk to you about foundry technology at Ginkgo for functional food proteins. Food proteins can be both diverse and complex, and functionality challenges make it even more difficult to address them when we're talking about organisms that make functional food proteins. Some of these functionalities are presented here on this slide. They could be nutritional functionality, such as primary amino acid profile. It could be the availability of cofactors or binding to cofactors. It could be immunogenic responses presented by glycosylation or antimicrobial activity. And there could be other factors which are sensory functionalities that go towards taste, such as thickening, stretch, and flavor profiles. And Ginkgo has worked in a lot of these areas and addressed several of these challenges through our full stack of foundry tools and technologies. These are complex problems that require simultaneous solutions to address productivity, titer goals, stability, and production challenges, processing challenges, while keeping the functionality intact. So today, I will present to you a few of these services that are offered by Ginkgo to address some of these challenges in a rapid and scalable way that can cater to your unique need. So before I go further, I just wanted to quickly ask, everyone's able to see my screen perfectly, right? A quick thumbs up will do.
You look great.
Perfect. All right, so let's start with DNA. So the very first building block, DNA, right? So Ginkgo's invested a lot in building our metagenomic database. We have both proprietary libraries, public and private databases with over 2 billion genes to access that we can comb through to provide you unique start points for addressing your unique need. Now, let's just give ourselves an example over here. You want an enzyme that's differentiated from what's out there in the market. You want a unique pathway to be debottlenecked or optimized. You want enzymes to provide post-translational modifications. You want homologs to scour through different sequences, whether it be in the protein space or in the enzyme space. Then discovery with metagenomics is a good start point. We have, like I said, over 2 billion genes to go through. Some of them sourced from known sources, others from microbes which have not yet been cultured. And this is a great start point to differentiate your protein and identify your start sequence. Now, in some cases, you may not want to change that start sequence. You may already know what you want to work with. In that case, the question comes, how do you want to improve that enzyme? How do you want to improve that protein, that sequence? Do you need to develop thermostability, tolerance states, acid tolerance, stability, reduce byproduct profile, reduce that off flavor, well, then enzyme evolution or engineering could be the next service that you could pick up. Ginkgo spent some time and effort in developing an automated AI platform, a unique AI tool called OWL, where we go through machine learning, combine that with our automation and data analytics so that we rapidly learn from each round of experimentation done in high throughput. We fine-tune our models, and then we essentially come up with that unique needle and haystack solution that can present the activity, the specific activity that you need, the functionality that you need, and also reduce the other activity that increases downstream processing challenges. So this is another offering. Now, those are about the primary sequence of the enzyme, right? How do we actually do our work in the foundry? Well, Ginkgo is essentially a combination of man and machine, right? We optimize our solutions, we optimize our scale, and we uniquely tailor and specific need. Our foundry is essentially our automation facility. We learn from the experiments that we run in this foundry, and that's our accumulated knowledge or code base. And all of this is done by our massive group of scientists and engineers. We have over 350 scientists and engineers, technical experts, who learn from and power the foundry so that we can continuously, repeatedly run design, build, test, and learn cycles. With our people, we are able to run in-house DNA synthesis, strain construction, high throughput strain culturing and testing, fermentations, and then develop know-how that can accelerate projects, such as how do you make your construct design? How do you recode for your protein of interest? How do we work with certain organisms so that they fit certain functionalities, regulatory requirements, and do it fast? So this is how we help optimize the bio to your need and at our scale. One example is the NCAPS technology, which basically is microfluidics on steroids, right? So you have microcolonies, which could come from various cell types. It could be bacteria, yeast, filamentous fungi, sometimes even plant cells, mammalian cells. And we can encapsulate these in nanoliter reactors, which are essentially beads. These would directly be screened through microfluidics, and we can screen up to 1 million clones per round, which essentially offers us the ability to screen and find that right productive strain. It will directly look for your protein readout or your enzyme readout, and also be able to look for growth metrics. So your strain's productivity is directly measured through the 1 million clone scan that we can run. It's an extremely versatile tool and could be used and extremely helpful for functional protein production screens. All right, so now let's take a step into the stars, our host organisms. Now, there can be several organisms that can be used for protein production, but particularly in the food space, we want to have low COGS, we want to have precision, we want to meet regulatory needs, we want to also achieve quick and rapid engineering so your solution can scale and hit the market early. One of our hosts is Pichia pastoris, or Comagatella fatia. And this organism has been rewired in our foundry in a way that it can essentially be tuned for expression using our synthetic expression systems. We've developed over 100,000 promoters, which are essentially regulated and tunable. Very important for functional protein production or enzyme production, because oftentimes growth needs to be decoupled from expression or production. This is completely methanol-free. We work with methanol systems, but our tunable expression systems are methanol-free systems. We've generated data on over 10,000 proteins in the food space using these expression systems. And some of our best titers are over 25 grams per liter of recombinant proteins from the mammalian world. These are validated for performance in high throughput. They go all the way from Amber 250s, where we can run media optimizations. We have scalable fermentations, and have been validated at over 10,000 liters for various food applications. Needless to say, our base strains, the synthetic expression systems, outperform the methanol standard best-in-class alcohol oxidase promoters by over 20-fold. But the important thing to remember here is, what does your protein need? it might not need the strongest promoter. It might need tunable expression. And that's what we are trying to achieve. All right, so the next one is Aspergillus niger. Why do we go into this host and why not stick with Pichia? Well, Pichia has its plus points. It's rapid, it scales well. But when we are talking about titers and titers beyond 25 gram per liter, you need a host that can meet that kind of protein turnover. And that's an Aspergillus strain, a filamentous fungus. So we invested in an Aspergillus niger strain, very unique in its strain type. Its phenotype is that it's low viscosity. You can see a picture of that strain over here on the plate. Almost looks like a yeast strain. We've engineered the strain to be devoid of proteases as far as possible, clean backgrounds. You can see it in the picture here with our STS page gels. And the strain has the capability to produce over 120 gram per liter of native protein and has been validated for several prototypes in the enzyme and protein world in the food space. We've then scaled down the Aspergillus niger system of filamentous fungus into our foundries, made it foundry compatible, and also generated fermentation processes going from amber 250 level all the way to liter scale. And we've done media optimizations with these strains, as in filamentous fungi, the process is as important as strain engineering itself. So these are essentially a set of tools that we've developed at Ginkgo, and we provide as service offerings to uniquely cater to your need in the food space. And with this, I think we are well placed to address the functional food protein market.
All right. Thank you so much. Thank you so much, Sneha. That was great. That was great. Now, for the next segment, we're gonna continue on this theme of Aspergillus. We want to go a little bit deeper into how some of these technologies get used in practice for real food protein engineering projects. And for this, I am very excited to welcome Peter Punt. Peter is a distinguished organism engineer in the Ginkgo Bioworks team in Zeist, the Netherlands. He has almost 40 years of experience in fungal biotechnology, being co-author on about 150 papers and patent applications. Peter also holds a guest professorship at Leiden University. Peter, welcome. Thank you so much for being here.
Yeah, thank you, Jake. So I'm very happy to tell the audience about our Aspergillus system in some more detail. And for that, I'll have a few slides to share with you. I will focus on a few aspects of the system, and they're given here in a way. The opportunity is Aspergillus has a long history of use in recombinant protein production and in protein production by itself. More than 100 years ago, the first processes were described. And the products that are made with the fungus have a safe status both in FDA and EFSA, and that's very important for a number of applications in regulatory systems. And, well, as was already mentioned, they have a high secretion capacity. But of course, there is no system that is only have good things. I mean, we say within Ginkgo that we make things easy to engineer, but that doesn't mean it's always easy to do that. And probably that's why you're also listening to this webinar. And the challenges that fungi have is that in the fermentation, because of the growth phenotype, you get a phenotype that is shown here in the movie in the bottom. And that is a typical industrial strain where the biomass accumulates and you get something like an applesauce. And that works. I mean, several tens of grams of protein can be produced in these systems, and some of these systems are just industrially used. But we have developed a strain, and that's in the top, which is the same fungus, but then a strain with mutations that makes it almost like a yeast culture, so very liquid. And that has a benefit because you can reach higher biomass titers with the same kind of oxygen supply. Having said that, because we have these different strains, we were also able to identify which genes make this phenotype. And so by doing that, we now have also the capability to engineer any strain, maybe your strain, to go to lower viscosity by specifically targetedly engineering the genes that we have identified. And the identification was done, it was not a simple thing. We had to combine classical genetics with strain crossings, with genome sequencing, with genome editing, but we made it and we found the responsible genes. It was a complex phenotype because morphology is a complex phenotype. That also brings me to the other aspect, because the other challenge in fungal research is the high throughput strain engineering is just not as high throughput as with yeast and bacteria. I mean, we have to be fair on that. But what was nice about, in a way, a lovely coincidence of the strain that we have developed was that this strain had some benefits in the whole process of strain engineering, because it grew as such small colonies on the plate. I mean, it grows slow on the plates, not in the fermenter, but because it grows that slow in a way, you can more easily manipulate in small scale setups like microtiter plates. So we have identified these low viscous strains, but they could also be used in the workflows that we are using within Ginkgo. And so I want to go to the next slide. What's happening? Oops. Okay. And so what we've done is we have this Aspergillus strain, and we've in a way adapted it to foundry scale engineering as we are doing that in Ginkgo, or also the vice versa. We have also adapted the foundry aspects to be amenable for the fungus to be used. So in a way, it's the four elements that you always see. So we can design genes to be expressing the protein of interest by having a library of proven genetic parts. We can then do the screening. We can then select the best strains and validate them in setups like in small-scale fermentations. And a typical result you get with that is like this. And what you see here is a couple of hundred different transformers that we obtained. And then in red are all the strains that are below the threshold of being positive in a certain protein. And the blue ones are all the positive ones. I mean, you see that the cutoff is maybe a bit arbitrary, but what you see also is that you see a lot of strains that are, let's say, mediocre in producing, but there are only a very few numbers, but they are really 10 or maybe even 20 times higher than what is the mean. And so that means that there is something like a numbers game going on with fungi. And that has to do with some of the aspects in the fungal biology. I don't want to go into too much of the details, but things like gene silencing, they can play a role in that. And so you need large numbers of transformants to be able to find the really positive outliers. Now, you can do that by hand and by colony picking, but here, in particular, this NCAPS technology really provides a unique opportunity to combine two unique aspects. I mean, the unique strains we have at Ginkgo and this unique screening technology, because by doing that, we can encapsulate what you see here in the top, a picture where the fungal strain is encapsulated in those nanobeads. And in this case, we use an enzyme assay, but can be different things. And what we can do is screen up a million variants per campaign. And the screens that the team in Basel, this team is in Basel, in Europe, can do is they can screen for growth. So you can screen only those things that are really growing well as being a transformant, or on enzyme activity, or a combined, and other clever screens can be developed. So that means that you can, from those million strains, you can down select to a couple of hundreds, and then that couple of hundred you can analyze, let's say, the traditional way I showed you on the previous slide. So that really unlocks the potential of large libraries for fungi. And what is nice about this technology that is not only amenable for GMO approaches, but also classical screens, because you don't need to do transformation. You can also do selection and using a biological selection to find your best strains. So this, I'm really very enthusiastic about the combination of these technologies, and we see the first, not only in the protein space, but also in small molecule space, of really very nice results there. So having said that, also want to show you a little bit on where we are, or what kind of enzymes and proteins you can think about in the food space or the feed space also. I mean, they're not that much different, animal feed or human food, different enzymes. I mentioned a number of different enzymes, but the last, let's say, 5 or 10 years, we also have been working more and more in non-enzymatic proteins. So also there we have a lot of proteins in the food space, and a few of them are mentioned here. And on the right side of this slide, I show you an example of something that we found, you could say more or less by coincidence. This is a result of a number of different food characteristics, so like gelling, foaming, emulsification, and that was obtained not by an animal protein, but in this case by a microbial protein. So we could produce a microbial protein, which has a safe status. I mean, it's derived from a grass organism, produced in a grass organism, and that has characteristics that are similar to the classical food proteins, like maybe egg proteins or milk proteins. So we're quite enthusiastic about that because that diversifies the opportunities in the food space even further. So if this has triggered your interest, well, of course, we are happy to talk with you about that further. And Jake, I give the word back to you.
All right. Thank you so much. Thank you so much, Peter. Now we're going to move from Aspergillus to Pichia. And when we were creating this event, we wanted to be careful. We don't think about Aspergillus and Pichia as being in competition. We love all of our children equally. And of course, we work with other microbes too. But Pichia and Aspergillus can be complementary. Between the two of them, they cover a lot of food space. So we do want to bring forward their relative strengths. And now it's Pichia's turn. And for this, we turn to Andrea Kamatari. Andrea is a senior director of organism engineering at Ginkgo Bioworks. He has 15 years of experience as a subject matter expert on industrial fermentation and biotechnology, strain development, genetic engineering, and small to large scale fermentation. Andrea, thank you so much for being here.
Thank you, Jake. It's great to be here. I'm happy to have the chance to share the work of our teams for our customers. And today, I will focus our attention on a very important class of proteins, which is iron-binding proteins. So, first of all, let me say a couple of words on why this is an important class. Well, let's start from the evolutionary perspective. So, iron actually provided the possibility for proteins to unlock new functionalities at some point during evolution. So proteins could do more. If we fast forward to modern age, as you can see in the box below, in fact, there is an iron deficiency, and that's a real issue. So people need more iron in their diet, and there is a large global demand for iron-rich food. We think that synthetic biology can help here, and Ginkgo can help as well. So, focusing our attention a little bit on the biology, iron is bound to proteins with three fundamental mechanisms. There are also others, but these are the three main ones. And in some cases, we have very complex pathways that generate the cofactors that bring iron to a protein. So here we have one of the first issues that we are tackling, which is complexity. Biology is hard. So how do we cope with the complexity that is given by the protein itself and the cofactor as well? A Ginkgo is shown on the next slide. So we have essentially four strategies to tackle this challenge at Ginkgo. Two of them are more generally related to protein expression at Ginkgo. My colleague showed very interesting aspects of this, so we'll go very briefly about that. Essentially, we have a patented optimized expression system, methanol-free, that relies on the synthetic circuit within the cell. We tackle the complexity of the protein itself by having a large combinatorial library of chaperones and other helping factors that help to shape the protein correctly. And protein folding is a big issue when it comes to protein production within Pichia. And then we have two more specific strategies that are related to iron-binding proteins. On one side, we want to have the best possible enzymes to generate the cofactor itself. And we had essentially, we use a metagenomic approach that Neha referred to at some point to identify the best possible enzymes to do that and combine them in a library to increase the titer of the cofactor itself. A sort of a collateral element to that, we also want the cofactor to reach our protein. And let's keep in mind that The challenge that we have is that we want to generate grams per liter of a protein. So the cofactor must cope with such a titer. And all these strategies are aimed to have a high titer of a fully loaded iron binding protein. So let's see very quickly what does this mean in terms of workflow at Ginkgo. So this is a pictorial representation of Ginkgo workflows. Our workflows are very iterative. Sneha mentioned that at some point with the DBTL cycle, and also very modular. So what we do is generally to start from a base strain. Let me add that when we say base strain, we actually refer to a very large portfolio of strains. Currently, we have approximately 300 different strains that have been specifically engineered to provide the best starting point for express your protein. And then using some of the strategies in this example, what we typically do is to generate an intermediate strain that is then validated under production condition. Now, if the titer is sufficient for our customer, great. Otherwise, we have other workflows that are more specific for strain development, where we take essentially the intermediate strain that is deriving from the previous workflow, and with more targeted modifications, we relieve basically the bottlenecks that we might have in order to, that prevents us to allow to have a titer that is required in this case. One other important element is that the modifications that we perform and in the strains that we generate are compatible with FDA regulations. So finally, I wanted to bring a quick example of showcasing a customer request for a protein that was obviously an iron-binding protein. So what we did at this point was to start from the strain that had a low titer. You can see this strain as one of the dots that is represented on the left side of this slide. So it's one of these guys over here. And by applying some of the strategies that I described, we were able to significantly increase the titer as measured as percentage of improvement from the base strain. So we had an intermediate strain at that point. We didn't feel it was enough, so we actually implemented some more specific changes to the strain that allowed us to generate a final strain that basically provided a titer that was more than 10 times the initial titer that we had, exceeding by 2 times the customer target. And we did that in approximately 6 months. Finally, one very important element is that this strain is under scale up right now.
All right. Thank you so much. Thank you so much, Andrea. Very cool stuff. Very cool. So this is the point in our program when hopefully you've seen some technology that you like and you're wondering, how do you get your project onto the Foundry? And for this, we have some news, some interesting new possibilities to explore that we're calling success-based pricing. And we'll turn to Alyssa Blaze. Alyssa is a director of growth here at Ginkgo. She has an MBA from Dartmouth University and now leads our commercial operations and strategy for enzymes. Welcome, Alyssa.
Thanks so much, Jake. All right, before I get into the details of success-based pricing, I want to review exactly what it is that you're buying. So just as a reminder here, we have 4 distinct services. Discovery, where we can find a wild type protein that meets your needs. Protein optimization, where we'll enhance a seed protein. Protein expression, where we'll improve your protein production. And then finally, fermentation and scale up, where we take and optimize a production process for commercial scale. Behind each of these services, we've built standardized workflows that utilize the best of our technology. And we are very excited to offer 3 of them using the success-based pricing framework. So you might be wondering why it is that we're doing this. Well, on this next slide here, I do have a graph that represents the breadth of work we've done just in enzymes in particular, and really just over the last few years, to give you a sense of what we do here at Ginkgo. So this graph on the y-axis, each of these rows is a top-level EC number. And on the x-axis are the number of tests we've run. Each of the smaller color blocks are a specific project. At the bottom, you see we have several stats here. We've run over 200 unique programs, completed over 60 million tests. Really, the point that I want to get across here is that we've done a lot of work in enzymes across a lot of different functional areas. And this allows us to feel very confident in our capabilities. So because of that, what we'd like to do is offer this success-based pricing framework that allows you to minimize your technical risk and also lower your financial exposure. So next, I'll talk a little bit about, what exactly this looks like. There are two components. The first is the R&D fee. And this is only triggered if we meet that technical success criteria that our team and your team will work together to set. This fee is really meant to cover the cost of our work, whether we're completing protein discovery or protein expression. And upon paying this fee, what you would get is a package that includes both results from the work that was completed, as well as a sample for you to test in your hands. The second component here is a one-time commercial fee. And this is triggered by your interest in obtaining commercial rights to whatever it is that we produce for you. You'll have opportunity, as I mentioned, to kind of evaluate this deliverable in your hands, and then you can decide whether you want to take that forward. The point of this fee is really to share in the value that we're creating for you. And it's meant to represent the entirety of the downstream value that we as Ginkgo would have. And upon paying, you would receive either ownership or exclusive rights, depending on what we're working on together. So all of this really, what we're trying to do is reduce the friction that you felt in getting onto our platform. All right, on this next slide, I'm going to talk a little bit more in detail about how the project using this framework might actually work. So let's talk about the inputs first. I mentioned this technical success criteria. You're probably wondering, how do we set this? What is it? So as your innovation partner, we are really looking to help to get you to your next research milestone. And so every technical success criteria is going to be unique to your needs. It's a custom process. We'll go through discussions, understand what your ultimate goals are, and then break down the problem so that we understand what needs to be achieved in each round of a service. For example, let's say you have an enzyme, and right now the enzyme optimum is at a pH of 7. You're looking to bring that down, let's say to 5. And that would allow you to lower some of your material costs. After conversing back and forth, we get to a point where we realize that within a round of our protein optimization service, achieving or finding a hit that has an optimum of 6 is really a milestone that moves the needle for you and gives you confidence in the ability to kind of move forward with research. So that's what we're talking about. We're trying to find something that is meaningful, helps you to get to your goals, and can be achieved in 1 or 2 rounds of one of these services that we've talked about. All right, so that's the technical success criteria setting. Other inputs could include an assay that will make high throughput here, the enzyme or the protein or the strain that we're working with, and just any other information that would be helpful as incoming tech transfer for us. We'd perform protein optimization service or one of the other services. And during the time that we're performing the service, we're communicating frequently with you around key junctures of that project. So you can expect kind of transparency and dialogue throughout the completion of that project. We'll get to the end, and there are two outcomes. One is we've achieved the success criteria. This would trigger the R&D fee, and we would share results in a sample with you. We'd talk with you about, in advance, how long you would need to evaluate the sample. So you'll have an evaluation period that works for you. And then at the end of that evaluation period, you would decide to opt in or opt out of the commercial rights. If you opt in, you'll pay and obtain the commercial rights. If you opt out, no fee is due. Alternatively, we could not meet the success criteria. In that case, we'll still share the results, but you do not owe the fee. We'll, of course, have some discussion around what has happened. At the end of that discussion, it might be that we got really close and you're still interested in this deliverable anyway, in which case we could move forward with evaluation and you'd kind of go through the sequences of activities in this first row. Alternatively, you could not be interested and we could close out the project and perhaps talk about what else we might work on together. So it's a little bit about how this would work in practice. I think just to summarize here, and the two critical criteria are that you're using one of these services that we talked about and that we're able to align on a technical success criteria in terms of project eligibility for using this pricing framework. But we're just really excited to be able to find this way to lower your risk and financial exposure, and look forward to having much more detailed conversations with many of you on this call. So thank you.
All right. Thank you so much, Alyssa. And that brings us to the Q&A sesh. And for this, I will direct your attention again to the Q&A feature, which is at the bottom of your Zoom window. You can type your questions in there. We'll do our best to answer either in writing or out loud. Some of you have already been using the feature. I love to see it. Fair question. How much does all this cost?
So let's talk about pricing from the context of what is needed to be done, right? I mean, we have different service offerings, and if it's discovery or enzyme engineering, the floor can be anywhere in the $100,000 to $300,000 range. It really depends on what the complexity of the protein or enzyme is. If it's about protein production, strain engineering, creating a strain that can be scaled for production, the dollar value can range from $500,000 all the way to a full $1 million, depending on the number of cycles of engineering we need to perform, the specific functionalities that we need to integrate into the strain's genome, and then process, right? So process development, scaling. So in all reality, it can be a very small body of work, which is crisp, packaged in the $200,000 to $300,000 range. It could be a large body of work, which is a connection of small packages of work, which can add up to $1 million or even be $500,000. We'd like to cater to your need. And this can be roughly estimated by choice of organism, complexity of protein, functionality needs, speed to market, any of these, right?
Yeah. Okay, that's fair. So yeah, it's hard tech. Of course, it depends, but that maybe gives people some sense of it. Okay, here's another one that I liked on the business side. I think this can be for you, Alyssa. How do we set the success criteria for a success-based payment? Who decides what success is?
Sure. So that is really a joint effort between both parties. Our technical teams will get together, really understand what that job to be done is for you. Internally, we'll discuss what we think could be possible within a cycle or two of our services, come back to you and kind of do a little bit of a back and forth. But I really want to emphasize that every challenge that comes to us is different. So it's hard to say, the success criteria will always look like X because it's going to be tailored to whatever your specific challenge is. But I would say, to summarize, really, it's something that is going to move the needle for you and help you de-risk your ultimate goal that we can achieve in 1 or 2 cycles of any given service.
Mm-hmm. Cool. Yeah, I think that's a great point. Yes. So all the technical challenges are different. I'm seeing a question here on the topic of technical challenges, so a more technical question. And this one I'll give to you, Peter. So the question is, how do you control growth of a filamentous fungus in an encapsulated environment? So this is for this ultra-high throughput NCAPS screening platform. How do you control growth inside these tiny little capsules? Are you referring only to your mutated strain, which doesn't form filaments, which grows small?
So it's a very good question. Of course, I mean we are lucky with that strain because it grows so compact. But we have done projects with say strains from our customers that have a completely different growth phenotype, and of course we can play with the environment in the beet. So we can play with nutrients, we can play with maybe even compounds that make growth more compact. And I didn't mention that, but the screening options are even wider than what we are talking about here, because we can also do co-encapsulation. For example, we can take reporter strains of a bacterium that responds to something that's produced by the fungus, and for that you don't need really very matured colonies. Even if it only produces small amounts, you can also detect it. You have to realize that every screen is as good as the screen, how you've built the screen. So when you have strains that produce different levels, then you can in a way calibrate the system. So it's relevant because you don't want the fungus to grow outside of the beet, because then you lose that clonality. But in practice, I think we can work around a lot of things. I mean, there will be maybe things that are incompatible, but the flexibility. I mean, that's why I'm so enthusiastic about the combination of fungal growth, which is completely different from yeast and bacterial growth, and these beets.
Another question with a bit of a technical angle. I'm not sure. This might be for you, Andrea. It might be for you, Sneha. The question is, how do you test for functionality? So if we're gonna make functional proteins, we're gonna need some kind of an assay for functionality. How do we approach that problem of testing for functionality? What kind of capabilities do we have? I'm looking at you to see who's sort of nodding more. Okay, Andrea, let's make that question for you.
Certainly. So functionality is very important. So typically, what happens is that during the assay development, if it's needed or required, we have a parallel pathway when we develop the assay or onboard assays from our customers that address this topic. Depending on the requirement for high throughput screening, we will have to tune up the throughput for such assays if functionality is an immediate requirement for during the screening phase. There are different flavors here, to be honest, and in general, the quickest and cheapest way to proceed about expression of our products relies on our established screening system, but that doesn't mean that we cannot implement functionality assays from day one to perform the screening like that. Not always is the cheapest option or the smartest option, but it's certainly a possibility. We certainly want to provide proteins that work to our customers, so that's a must.
That's great. They love it. They love it when the proteins work. Absolutely. All right. I think we have time for one more question, and I'll do this one on the business side. I'm seeing various questions coming in on the topic of our track record in the food space, our experience in the food space. Who have you already worked with in the food proteins area? What are your established collaborations in food proteins? Who's feeling that one? Is that an Alyssa question? Is it a Steve question?
Not for me.
Alyssa, go for it. Or
go for it, Alyssa.
You might be better suited for that one, Sneha.
Okay, all right. So in the food space, it's public information that I can provide. We can only talk about that. So we have recently, we have our ongoing collaboration with Motif. for their products. And then we've also recently launched public announcements with Voodoo, as well as Ambrosia Bio in the sweetener space. So those are three public announcements that I can point towards, and we have others that we cannot talk about right now.
Yeah.
So
I think I like the Voodoo and the Ambrosia partnerships. Those were announced. I think those are sort of a good example of companies that are using our enzyme and protein services in creative ways. And I would direct people to that information. All right. So let's see here. Do we have any more sort of lightning round? I'm seeing we've still got 12 open questions, so we'll be able to follow up with people offline on these, because I think that the time has come to wrap up this virtual event. Thank you so much for joining us. Thank you so much for joining us. Some highlights from today, some take-home messages. The strain can make all the difference. Foundry scale engineering can take all the difference. Today's call to action is going to be nutrition wellness at ginkgobioworks.com. If you're ready to get on the foundry, operators are standing by. Put that $500 million worth of Ginkgo Foundry infrastructure to work on your food protein project. Thank you so much to the team who came out today. That's Sneha, Peter, Andrea, Alyssa, and Steve. Until next time, as we like to say around here at Ginkgo, let's grow.